Okay, picking up where we left off, the last slide we discussed in class on Wednesday was this, which is the end of the discussion of carbohydrates. Okay. The next macromolecule we're going to talk about are the lipids. And so these, in terms of the function and how you're going to see them in cells, one of the main functions is basically long-term energy storage. We know that if you eat a lot of sugary foods, a lot of carbs, your body tends to metabolize that into fats. Again, that is because it's just a better, you can pack more energy into a gram of lipids than you can a gram of carbohydrates in your body. Glycogen, that big polysaccharide that we use in our body, is more of a short-term storage. If the body thinks that it needs to take those calories and put them away for the long term, it will do so in the form of lipid fats. Insulation is also a use for animals and plants. A lot of hormones are built out of, we're going to talk about steroids and about how they're built from these type of lipids. And then the other big thing is these phospholipids that are basically the component of the building blocks of our cell membrane. These are the types of fats we're going to talk about. Okay, so polymer, lipid, monomers, these are the little components of lipids. Right? Basically, lipids are made up of glycerol and fatty acids, which you see here. Here's this plus this, right? We're taking the hydroxide off of the fatty acid, hydrogen from the glycerol. So this is dehydration synthesis, producing this big type of fat on the bottom. Okay, a couple things to notice in here is the very non-polar nature of this molecule. There are some oxygen in here. With these long, we call them hydrocarbon tails. Basically, a bunch of carbons and hydrogens attached on there. That is going to really make this a non-polar molecule. Okay. Other thing on this slide to note, ester linkage is just a fancy term for the type of covalent bond that is formed between the glycerol and fatty acid. I didn't really talk about this last time, but for the for the carbohydrates, the type of covalent bond formed between monosaccharides they're called glycosidic linkages. Okay, so it's just if you had a Venn diagram, the big term would be chemical bonds. The middle one, smaller one, would be covalent or ionic, and then inside of covalent bond, we have glycosidic linkages, and now we're talking about ester linkages. Okay. Saturated and unsaturated fats. So looking at these hydrocarbon tails, if all of the carbons in the tail are have two hydrogen covalently bonded to them, it's called a saturated fatty acid. These are more related to often animal fats. So you think of like we just had bacon last night. Think of that nasty fat in the bottom of the pan that is it basically solidifies when it cools. That's because it's saturated and these things are not branched and they tend to just lay in really dense, so it makes it a solid. Unsaturated fats, on the other hand, we see a double covalent bond between these two carbons, meaning that there's two fewer hydrogen, and really this usually takes on a bent shape, because the individual fatty acids or the hydrocarbon tails are not straight and they're branched. They can't fit together like this. There's a little space between them, and so they tend to be liquid at room temperature. Essential fatty acids. trying to think what to tell you to know about these. Is basically this part of the molecule shown going out to the COOH. That is the hydrocarbon tail. Right? That is a bunch of carbons and hydrogens. So that will tell you that this is a lipid. These are types of fats that your body cannot produce, right? And so they must be gained through your diet. So that's why doctors recommend these types of fish if they're high essential fatty acids. And it's ironic, but eating these fats actually reduce your risk of heart disease and reduce the bad fats in the body. Waxes are another type of lipid that are common in the plant and animal world. Going with the theme here, these are hydrophobic molecules, and they basically are on the leaf to keep water in and make them watertight. Other biologically important lipids. This is a big one, phospholipids. So this molecule, we call it amphipathic. Means it's sort of like, I guess, analogous to polar, in that the individual components of the macromolecule have different behaviors in terms of whether they're hydrophilic or hydrophobic. So in this case, the head part of this, which is made up of the glycerol and phosphate, are hydrophilic, and you can see that's because of this negative charge on the oxygen, right? So it has a polar head and it has non-polar tails. These tails are just long hydrocarbons. The black little units here are carbon, and the white units are hydrogen, and so the tails are nothing but hydrocarbon tails. Okay. And these are hydrophobic because those are hydrophobic and these are hydrophilic. If you put a bunch of phospholipids into water, what is supposed to happen? The heads will stick together, right? The tails will stick together, and because the tails don't like the water, they're going to turn one way away from the water, and then another layer of phospholipids will form, and basically the tails will point towards each other to keep away from the water. And so this basically is exactly what a cell membrane is. It's a phospholipid bilayer. So your cells all have basically like let's say it's the consistency of olive oil, a layer of lipids surrounding each one of your cells. Steroids, see the ring shape, the ring structure. These are precursors to a lot of vitamins and a lot of hormones. So I would be able to recognize the structure of these and know where you would find them. Right? This is a cholesterol, vitamin D, and these important hormones in the body. Okay, so that's really all I want to say about the lipids. The next major type of macromolecule, actually I'm going to cut it off.